So this is an electron microscopy figure showing a limb bud, and this is the AER present at the most distal site of the limb bud between the dorsal and the ventral ectoderm. When the dorsal ectoderm and the ventral ectoderm will meet up, they will form this kind of structure called AER, which is a signaling center for the formation of the limbs according to the proximal distal axis. So the roles of the AER is to maintain the mesenchyme beneath in a proliferating state in order to keep the limb growing according to the proximal distal axis. Also, it maintains the expression of molecules that generate the anterior posterior axis, as I explained before. We have communication between FGF8 in the AER and other molecules in the ZPA, the signaling center important for the anterior posterior growth of the limbs. And we have interaction between the proteins specifying the anterior posterior axis and dorsal ventral axis, so that each cell will be given instructions on how to differentiate into a very specific way during the limbs formation according to this interaction, and the AER will control this kind of interaction between these different proteins. So here I'm showing you again the roles of FGF10 versus FGF8 in the formation of the limb according to the proximal distal axis. So FGF10 is present between the dorsal and the ventral side of the embryo, dorsal and ventral ectoderm, and the boundary when dorsal and ventral ectoderm meet up will form AER, as I explained in the figure before. So in chicken embryos having mutations so that they cannot develop ventral side, they are dorsalized, so there will be no dorsal versus ventral junction in the limb bud. These chicken mutants are limbless, they cannot develop limbs because the AER fail to form in this case and the limb development will be stopped. Okay, so no AER, no limbs formation. So FGF10 usually, as we mentioned before, is released by the lateral plate mesoderm. Okay, and it acts by inducing Wnt protein, like Wnt protein to activate beta-catenin here in the ectoderm. So for example, we have Wnt3A in chicken and Wnt3 in humans and mice. So we have conservation of Wnt induced by the FGF10. This Wnt is present in the ectoderm, as I mentioned before. So this Wnt will act through the canonical beta-catenin pathway to induce FGF8 right here in the ectoderm in order to form the AER. Okay, so this FGF8 in the AER will stimulate back, FGF8 here will stimulate back the proliferation of the mesenchyme by keeping the expression of FGF10 in this lateral plate mesoderm on all the time. Okay, so we have a kind of positive feedback loop between the mesenchyme and the overlying ectoderm. So the mesenchyme will produce FGF10. FGF10 act on the activation of FGF8 in the ectoderm or the AER, and the FGF8 in the AER will maintain the expression of FGF10 in the mesenchyme and this positive feedback loop will maintain the growth of the limb bud according to the proximal distal axis. So we talked that AER is very important for the formation of the proximal distal limb, or for the proximal distal growth for the limb. Here we can see the skeleton of a normal chick wing. Okay, and here in B we can see views of the skeletal pattern of this chicken wing after the removal of the AER okay, at different stages during development. For example, when they removed it very early, the stylopod just is formed a bit, and then we have a stop in the formation or the development of the limb. Okay, if we remove it a bit later, if we remove the AR a bit later, we have more elongation of the stylopod. If we remove it also later, sometimes later during development, we can see the stylopod is formed, and we have the zeugopod, a bit of it is formed. And here we have it, we have two bones because we made the AER removal later than before. So according to these experiments, we can conclude that the AER structure is highly important and necessary for wing development in chicken. No AER, no wing development. Okay? And when we remove the AER, the limb development will be stopped at the location of the bone already formed. And now I will explain you different experiments showing the importance of the AER during the limb growth. For example, here in this first experiment, we have a limb bud. This is the AER, and beneath the AER we have a four-limb mesenchyme. So we expect the formation, in this case, of a forelimb from a limb bud. So if we remove, for example, the AER in the first experiment, we see that the limb development will stop immediately. Okay, it will stop at the moment of its removal. Here in this case we have already the stylopod, and then it stops because we removed it at this time. So if we graft, for example, one extra AER just around the first AER, in this case we can see that the wing is duplicated. We have a duplicated wing digits. If we, for example, replace the forelimb mesenchyme with a leg mesenchyme, okay, in this case we can get legs or leg bones induced by the leg mesenchyme. Okay, we can see that we have formation of legs at the most distal part because these are induced by the AER, the leg, by the leg mesenchyme. Okay, that will form the leg bones. Okay, knowing that we started here with the formation of a wing before the replacement of the forelimb mesenchyme by a leg mesenchyme. Okay, but let's say we replace the forelimb mesenchyme with a non-limb mesenchyme. It means with a heart mesenchyme, with a skin mesenchyme, with a brain mesenchyme, anything different than the limb mesenchyme. In this case also we get similar result as the first, as if we remove the AER. Okay, so the AER in this case will regress and the limb development will stop. So according to this experiment we have seen, okay, the first four experiments, we can see that we'll need a specific interaction between the AER that should be present and the mesenchyme beneath that should be specific for the limb, and this kind of interaction between limb mesenchyme and AER is essential to induce the formation of the limb. In the last experiment they just removed the AER and they replaced this AER by beads called FGF beads, so beads able to produce FGF. In this case it should be FGF8 instead of the AER. And in this case we got a normal wing development. So what does it mean? From this experiment we conclude that the FGF8 is a main component of the AER. If it can replace the AER itself because it's the main component and the main molecule responsible for the activity of the AER signaling center. And in this case, if the ectoderm is producing FGF8, the underlying mesenchyme, the forelimb mesenchyme in blue, will produce FGF10. So we'll keep having this loop FGF8, FGF10 in order to produce a normal wing. And I just want to mention regarding the limb bud, we have the AER at the most distal side. Just beneath the AER, we have a region of the mesenchyme, the one in green, called progress zone. It's a zone of high mitotic activity, and usually the cells are highly proliferating here in order to increase the size of the limb during its growth. Here in this experiment, we can see that they grafted at this location the progress zone of an early wing bud. They take this progress zone, the one we have seen in green, from an early wing bud, and they transplanted it in a late wing bud that already formed, you see, ulna and radius, okay, humerus, ulna and radius, and they grafted this wing bud progress zone right here, and we can see that the limb or the limb grew up from the end point, so it means it will continue, okay, by forming stylopod, ulna, radius, and finally the otopod. So it means it starts from the beginning, okay, at the point of the grafting it started from the beginning to form the stylopod, ulna, radius, and the zygopod. Since this interaction between AER and the progress zone will happen at this point of grafting to promote from de novo, from the beginning, a formation of new limb. So we can see that the proximal distal growth is made up by interactions between the AER and the limb mesenchyme directly.